Fear. Published by Wikipedia. Fear is a feeling induced by perceived danger or threat that occurs in certain types of organisms, which causes a change in metabolic and organ functions and ultimately a change in behavior, such as fleeing, hiding, or freezing from perceived traumatic events. Fear in human beings may occur in response to a certain stimulus occurring in the present, or in anticipation or expectation of a future threat perceived as a risk to body or life. The fear response arises from the perception of danger leading to confrontation with or escape from avoiding the threat, which in extreme cases of fear can be a freeze response or paralysis. In humans and animals, fear is modulated by the process of cognition and learning. Thus, fear is judged as rational or appropriate and irrational or inappropriate. An irrational fear is called a phobia. Psychologists such as John B. Watson, Robert Plutchik, and Paul Ekman have suggested that there is only a small set of basic or innate emotions and that fear is one of them. This hypothesized set includes such emotions as acute stress reaction, anger, angst, anxiety, fright, horror, joy, panic, and sadness. Fear is closely related to, but should be distinguished from, the emotion anxiety, which occurs as the result of threats that are perceived to be uncontrollable or unavoidable. The fear response serves survival by engendering appropriate behavioral responses, so it has been preserved throughout evolution. Sociological and organizational research also suggests that individuals' fears are not solely dependent on their nature but are also shaped by their social relations and culture, which guide their understanding of when and how much fear to feel. Signs and symptoms. Many physiological changes in the body are associated with fear, summarized as the fight or flight response. An inborn response for coping with danger, it works by accelerating the breathing rate, heart rate, vasoconstriction of the peripheral blood vessels leading to blushing and sanskadania of the central vessels, increasing muscle tension including the muscles attached to each hair follicle to contract and causing goosebumps, or more clinically, piloerection, sweating, increased blood glucose, increased serum calcium, increase in white blood cells called neutrophilic leukocytes, alertness leading to sleep disturbance and butterflies in the stomach. This primitive mechanism may help an organism survive by either running away or fighting the danger. With the series of physiological changes, the consciousness realizes an emotion of fear. Causes. People develop specific fears as a result of learning. This has been studied in psychology as fear conditioning, beginning with John B. Watson's Little Albert experiment in 1920, which was inspired after observing a child with an irrational fear of dogs. In this study, an 11-month-old boy was conditioned to fear a white rat in the laboratory. The fear became generalized to include other white, furry objects, such as a rabbit, dog, and even a ball of cotton. Fear can be learned by experiencing or watching a frightening traumatic accident. For example, if a child falls into a well and struggles to get out, he or she may develop a fear of wells, heights, enclosed spaces, or water. There are studies looking at areas of the brain that are affected in relation to fear. When looking at these areas, it was proposed that a person learns to fear regardless of whether they themselves have experienced trauma or if they have observed the fear in others. In a study completed by Andreas Olson, Catherine I. Nearing and Elizabeth A. Phelps, the amygdala were affected both when subjects observed someone else being submitted to an aversive event, knowing that the same treatment awaited themselves, and when subjects were subsequently placed in a fear-provoking situation. This suggests that fear can develop in both conditions, not just simply from personal history. Fear is affected by cultural and historical context. For example, in the early 20th century, many Americans feared polio, a disease that can lead to paralysis. There are consistent cross-cultural differences in how people respond to fear. Display rules affect how likely people are to show the facial expression of fear and other emotions. Emotions of fear could be also influenced by gender. Research has shown participants were able to recognize the facial expression of fear significantly better on a male face than on a female face. Females also recognized fear generally better than males. Although many fears are learned, the capacity to fear is part of human nature. Many studies have found that certain fears are much more common than others. These fears are also easier to induce in the laboratory. This phenomenon is known as preparedness. Because early humans that were quick to fear dangerous situations were more likely to survive and reproduce, preparedness is theorized to be a genetic effect that is the result of natural selection. From an evolutionary psychology perspective, different fears may be different adaptations that have been useful in our evolutionary past. They may have developed during different time periods. Some fears, such as fear of heights, may be common to all mammals and developed during the Mesozoic period. Other fears, such as fear of snakes, may be common to all simians and developed during the Cenozoic time period. Still others, such as fear of mice and insects, may be unique to humans and developed during the Paleolithic and Neolithic time periods. Fear is high only if the observed risk and seriousness both are high, and it is low if risk or seriousness is low. Top 10 types in the U.S. In a 2005 Gallup poll, a national sample of adolescents between the ages of 13 and 17 were asked what they feared the most. The question was open-ended and participants were able to say whatever they wanted. The top 10 fears were, in order, terrorist attacks, spiders, death, failure, war, criminal or gang violence, being alone, the future, and nuclear war. In an estimate of what people fear the most, book author Bill Tanzer analyzed the most frequent online queries that involve the phrase, fear of, following the assumption that people tend to seek information on the issues that concern them the most. His top 10 list of fears published 2008 consisted of flying, heights, clowns, intimacy, death, rejection, people, snakes, failure, and driving. Common phobias. According to surveys, some of the most common fears are of demons and ghosts, the existence of evil powers, cockroaches, spiders, snakes, heights, trypophobia, water, enclosed spaces, tunnels, bridges, needles, social rejection, failure, examinations, and public speaking. Fear of death. Death anxiety is multidimensional. It covers fears related to one's own death, the death of others, fear of the unknown after death, fear of obliteration, and fear of the dying process, which includes fear of a slow death and a painful death. Death anxiety is one's uncertainty to dying. However, there is a more severe form of having a fear of death, which is known as thanatophobia, which is anxiety over death that becomes debilitating or keeps a person from living their life. The Yale philosopher Shelley Kagan examined fear of death in a 2007 Yale Open course by examining the following questions: is fear of death a reasonable, appropriate response? What conditions are required and what are appropriate conditions for feeling fear of death? What is meant by fear, and how much fear is appropriate? According to Kagan, for fear in general to make sense, 3 conditions should be met. The object of fear needs to be something bad. There needs to be a non-negligible chance that the bad state of affairs will happen. There needs to be some uncertainty about the bad state of affairs. The amount of fear should be appropriate to the size of the bad. If the 3 conditions are not met, fear is an inappropriate emotion. He argues that death does not meet the first 2 criteria, even if death is a deprivation of good things and even if one believes in a painful afterlife. Because death is certain, it also does not meet the 3rd criterion, but he grants that the unpredictability of when one dies may be cause to a sense of fear. In a 2003 study of 167 women and 121 men, aged 65 to 87, low self-efficacy predicted fear of the unknown after death and fear of dying for women and men better than demographics, social support, and physical health. Fear of death was measured by a multidimensional fear of death scale, which included the 8 subscales: fear of dying, fear of the dead, fear of being destroyed, fear for significant others, fear of the unknown, fear of conscious death, fear for the body after death, and fear of premature death. In hierarchical multiple regression analysis, the most potent predictors of death fears were low spiritual health efficacy, defined as beliefs relating to one's perceived ability to generate spiritually based faith and inner strength, and low instrumental efficacy, defined as beliefs relating to one's perceived ability to manage activities of daily living. Psychologists have tested the hypotheses that fear of death motivates religious commitment, and that assurances about an afterlife alleviate the fear. However, empirical research on this topic has been equivocal. Religiosity can be related to fear of death when the afterlife is portrayed as time of punishment. Intrinsic religiosity, as opposed to mere formal religious involvement, has been found to be negatively correlated with death anxiety. In a 1976 study of people of various Christian denominations, those who were most firm in their faith, who attended religious services weekly, were the least afraid of dying. The survey found a negative correlation between fear of death and religious concern. In a 2006 study of white, Christian men and women, the hypothesis was tested that traditional, church-centered religiousness and deinstitutionalized spiritual seeking are ways of approaching fear of death in old age. Both religiousness and spirituality were related to positive psychosocial functioning, but only church-centered religiousness protected subjects against the fear of death. Fear of the unknown. Fear of the unknown or irrational fear is caused by negative thinking which arises from anxiety accompanied with a subjective sense of apprehension or dread. Irrational fear shares a common neural pathway with other fears, a pathway that engages the nervous system to mobilize bodily resources in the face of danger or threat. Many people are scared of the unknown. The irrational fear can branch out to many areas such as the hereafter, the next 10 years, or even tomorrow. Chronic irrational fear has deleterious effects since the elicitor stimulus is commonly absent or perceived from delusions. Such fear can create comorbidity with the anxiety disorder umbrella. Being scared may cause people to experience anticipatory fear of what may lie ahead rather than planning and evaluating for the same. For example, continuation of scholarly education is perceived by many educators as a risk that may cause them fear and stress, and they would rather teach things they've been taught than go and do research. That can lead to habits such as laziness and procrastination. The ambiguity of situations that tend to be uncertain and unpredictable can cause anxiety in addition to other psychological and physical problems in some populations, especially those who engage it constantly, for example, in war-ridden places or in places of conflict, terrorism, abuse, etc. Poor parenting that instills fear can also debilitate a child's psyche development or personality. For example, parents tell their children not to talk to strangers in order to protect them. In school, they would be motivated to not show fear in talking with strangers, but to be assertive and also aware of the risks and the environment in which it takes place. Ambiguous and mixed messages like this can affect their self-esteem and self-confidence. Researchers say talking to strangers isn't something to be thwarted but allowed in a parent's presence if required. Developing a sense of equanimity to handle various situations is often advocated as an antidote to irrational fear and as an essential skill by a number of ancient philosophies. Fear of the unknown may be or possibly the, fundamental fear. Mechanism. Often laboratory studies with rats are conducted to examine the acquisition and extinction of conditioned fear responses. In 2004, researchers conditioned rats to fear a certain stimulus through electric shock. The researchers were able to then cause an extinction of this conditioned fear, to a point that no medications or drugs were able to further aid in the extinction process. However, the rats did show signs of avoidance learning, not fear, but simply avoiding the area that brought pain to the test rats. The avoidance learning of rats is seen as a conditioned response, and therefore the behavior can be unconditioned, as supported by the earlier research. Species-specific defense reactions or avoidance learning in nature is the specific tendency to avoid certain threats or stimuli. It is how animals survive in the wild. Humans and animals both share these species-specific defense reactions, such as the flight or fight, which also includes pseudo-aggression, fake or intimidating aggression, and freeze response to threats, which is controlled by the sympathetic nervous system. These SSDRs are learned very quickly through social interactions between others of the same species, other species, and interaction with the environment. These acquired sets of reactions or responses are not easily forgotten. The animal that survives is the animal that already knows what to fear and how to avoid this threat. An example in humans is the reaction to the sight of a snake. Many jump backwards before cognitively realizing what they are jumping away from, and in some cases it is a stick rather than a snake. As with many functions of the brain, there are various regions of the brain involved in deciphering fear in humans and other non-human species. The amygdala communicates both directions between the prefrontal cortex, hypothalamus, the sensory cortex, the hippocampus, thalamus, septum, and the brainstem. The amygdala plays an important role in SSDR, such as the ventral amygdalofugal, which is essential for associative learning, and SSDRs are learned through interaction with the environment and others of the same species. An emotional response is created only after the signals have been relayed between the different regions of the brain, and activating the sympathetic nervous systems, which controls the flight, fight, freeze, fright, and faint response. Often a damaged amygdala can cause impairment in the recognition of fear. This impairment can cause different species to lack the sensation of fear, and often can become overly confident, confronting larger peers, or walking up to predatory creatures. Robert C. Bowles, a researcher at University of Washington, wanted to understand species-specific defense reactions and avoidance learning among animals, but found that the theories of avoidance learning and the tools that were used to measure this tendency were out of touch with the natural world. He theorized the species-specific defense reaction. There are three forms of SSDRs: flight, fight, or freeze. Even domesticated animals have SSDRs, and in those moments it is seen that animals revert to atavistic standards and become wild again. Dr. Bowles states that responses are often dependent on the reinforcement of a safety signal and not the aversive conditioned stimuli. This safety signal can be a source of feedback or even stimulus change. Intrinsic feedback or information coming from within, muscle twitches, increased heart rate, are seen to be more important in SSDRs than extrinsic feedback, stimuli that comes from the external environment. Dr. Bowles found that most creatures have some intrinsic set of fears to help assure survival of the species. Rats will run away from any shocking event, and pigeons will flap their wings harder when threatened. The wing flapping in pigeons and the scattered running of rats are considered species-specific defense reactions or behaviors. Bowles believed that SSDRs are conditioned through Pavlovian conditioning and not operant conditioning. SSDRs arise from the association between the environmental stimuli and adverse events. Michael S. Fancillo conducted an experiment to test some specific defense reactions. He observed that rats in two different shock situations responded differently based on instinct or defensive topography rather than contextual information. Species-specific defense responses are created out of fear and are essential for survival. Rats that lack the gene Stathman show no avoidance learning or a lack of fear and will often walk directly up to cats and be eaten. Animals use these SSDRs to continue living, to help increase their chance of fitness by surviving long enough to procreate. Humans and animals alike have created fear to know what should be avoided, and this fear can be learned through association with others in the community, or learned through personal experience with a creature, species, or situations that should be avoided. SSDRs are an evolutionary adaptation that has been seen in many species throughout the world, including rats, chimpanzees, prairie dogs, and even humans, an adaptation created to help individual creatures survive in a hostile world. Fear learning changes across the lifetime due to natural developmental changes in the brain. This includes changes in the prefrontal cortex and the amygdala, neurocircuit in mammals. The thalamus collects sensory data from the senses. Sensory cortex receives data from the thalamus and interprets it. Sensory cortex organizes information for dissemination to the hypothalamus, amygdala, hippocampus. The brain structures that are the center of most neurobiological events associated with fear are the two amygdalae, located behind the pituitary gland. Each amygdala is part of a circuitry of fear learning. They are essential for proper adaptation to stress and specific modulation of emotional learning memory. In the presence of a threatening stimulus, the amygdalae generate the secretion of hormones that influence fear and aggression. Once a response to the stimulus in the form of fear or aggression commences, the amygdalae may elicit the release of hormones into the body to put the person into a state of alertness, in which they are ready to move, run, fight, etc. This defensive response is generally referred to in physiology as the fight or flight response, regulated by the hypothalamus, part of the limbic system. Once the person is in safe mode, meaning that there are no longer any potential threats surrounding them, the amygdala will send this information to the medial prefrontal cortex, where it is stored for similar future situations, which is known as memory consolidation. Some of the hormones involved during the state of fight or flight include epinephrine, which regulates heart rate and metabolism as well as dilating blood vessels and air passages, norepinephrine increasing heart rate, blood flow to skeletal muscles and the release of glucose from energy stores, and cortisol, which increases blood sugar, increases circulating neutrophilic leukocytes, calcium, amongst other things. After a situation which incites fear occurs, the amygdala and hippocampus record the event through synaptic plasticity. The stimulation to the hippocampus will cause the individual to remember many details surrounding the situation. Plasticity and memory formation in the amygdala are generated by activation of the neurons in the region. Experimental data supports the notion that synaptic plasticity of the neurons leading to the lateral amygdala occurs with fear conditioning. In some cases, this forms permanent fear responses such as post-traumatic stress disorder or a phobia. MRI and fMRI scans have shown that the amygdala in individuals diagnosed with such disorders, including bipolar or panic disorder, are larger and wired for a higher level of fear. Pathogens can suppress amygdala activity. Rats infected with the toxoplasmosis parasite become less fearful of cats, sometimes even seeking out their urine-marked areas. This behavior often leads to them being eaten by cats. The parasite then reproduces within the body of the cat. There is evidence that the parasite concentrates itself in the amygdala of infected rats. In a separate experiment, rats with lesions in the amygdala did not express fear or anxiety towards unwanted stimuli. These rats pulled on levers supplying food that sometimes sent out electrical shocks. While they learned to avoid pressing on them, they did not distance themselves from these shock-inducing levers. Several brain structures other than the amygdala have also been observed to be activated when individuals are presented with fearful versus neutral faces, namely the occipitos rebeller regions including the fusiform gyrus and the inferior parietal, superior temporal gyri. Fearful eyes, brows and mouth seem to separately reproduce these brain responses. Scientists from Zurich studies show that the hormone oxytocin related to stress and sex reduces activity in your brain fear center. Pheromones and why fear can be contagious. In threatening situations, insects, aquatic organisms, birds, reptiles, and mammals emit odorant substances, initially called alarm substances, which are chemical signals now called alarm pheromones. This is to defend themselves and at the same time to inform members of the same species of danger and leads to observable behavior change like freezing, defensive behavior, or dispersion depending on circumstances and species. For example, stressed rats release odorant cues that cause other rats to move away from the source of the signal. After the discovery of pheromones in 1959, alarm pheromones were first described in 1968 in ants and earthworms, and 4 years later also found in mammals, both mice and rats. Over the next 2 decades, identification and characterization of these pheromones proceeded in all manner of insects and sea animals, including fish, but it was not until 1990 that more insight into mammalian alarm pheromones was gleaned. Earlier, in 1985, a link between odors released by stressed rats and pain perception was discovered. Unstressed rats exposed to these odors developed opioid-mediated analgesia. In 1997, researchers found that bees became less responsive to pain after they had been stimulated with isoamyl acetate, a chemical smelling of banana, and a component of bee alarm pheromone. The experiment also showed that the bees' fear-induced pain tolerance was mediated by an endorphin. By using the force swimming test in rats as a model of fear induction, the first mammalian alarm substance was found. In 1991, this alarm substance was shown to fulfill criteria for pheromones: well-defined behavioral effect, species specificity, minimal influence of experience and control for nonspecific arousal. Rat activity testing with the alarm pheromone and their preference avoidance for odors from cylinders containing the pheromone showed that the pheromone had very low volatility. In 1993, a connection between alarm chemosignals in mice and their immune response was found. Pheromone production in mice was found to be associated with or mediated by the pituitary gland in 1994. In 2004, it was demonstrated that rats' alarm pheromones had different effects on the recipient rat depending which body region they were released from. Pheromone production from the face modified behavior in the recipient rat, e.g., caused sniffing or movement, whereas pheromones secreted from the rat's anal area induced autonomic nervous system stress responses, like an increase in core body temperature. Further experiments showed that when a rat perceived alarm pheromones, it increased its defensive and risk assessment behavior, and its acoustic startle reflex was enhanced. It was not until 2011 that a link between severe pain, neuroinflammation, and alarm pheromones release in rats was found. Real-time RT-PCR analysis of rat brain tissues indicated that shocking the footpad of a rat increased its production of pro-inflammatory cytokines in deep brain structures, namely of IL-1 beta, heteronuclear corticotropin-releasing hormone, and CFOS mRNA expressions in both the paraventricular nucleus and the bed nucleus of the stria terminalis, and it increased stress hormone levels in plasma. The neurocircuit for how rats perceive alarm pheromones was shown to be related to the hypothalamus, brainstem, and amygdala, all of which are evolutionary ancient structures deep inside or in the case of the brainstem underneath the brain away from the cortex, and involved in the fight or flight response, as is the case in humans. Alarm pheromone-induced anxiety in rats has been used to evaluate the degree to which anxiolytics can alleviate anxiety in humans. For this, the change in the acoustic startle reflex of rats with alarm pheromone-induced anxiety has been measured. Pretreatment of rats with one of 5 anxiolytics used in clinical medicine was able to reduce their anxiety, namely midazolam, phenelzine, propranolol, a non-selective beta blocker, clonidine, an alpha-2 adrenergic agonist or CP154,526, a corticotropin-releasing hormone antagonist. Faulty development of odor discrimination impairs the perception of pheromones and pheromone-related behavior, like aggressive behavior and mating in male rats. The enzyme mitogen-activated protein kinase 7 has been implicated in regulating the development of the olfactory bulb and odor discrimination, and it is highly expressed in developing rat brains, but absent in most regions of adult rat brains. Conditional deletion of the MAPK7 gene in mouse neural stem cells impairs several pheromone-mediated behaviors, including aggression and mating in male mice. These behavior impairments were not caused by a reduction in the level of testosterone, by physical immobility, by heightened fear or anxiety, or by depression. Using mouse urine as a natural pheromone-containing solution, it has been shown that the impairment was associated with defective detection of related pheromones and with changes in their inborn preference for pheromones related to sexual and reproductive activities. Lastly, alleviation of an acute fear response because a friendly peer tends and befriends is called social buffering. The term is in analogy to the 1985 buffering hypothesis in psychology, where social support has been proven to mitigate the negative health effects of alarm pheromone-mediated distress. The role of a social pheromone is suggested by the recent discovery that olfactory signals are responsible in mediating the social buffering in male rats. Social buffering was also observed to mitigate the conditioned fear responses of honeybees. A bee colony exposed to an environment of high threat of predation did not show increased aggression and aggressive-like gene expression patterns in individual bees, but decreased aggression. That the bees did not simply habituate to threats is suggested by the fact that the disturbed colonies also decreased their foraging. Biologists have proposed in 2012 that fear pheromones evolved as molecules of keystone significance, a term coined in analogy to keystone species. Pheromones may determine species compositions and affect rates of energy and material exchange in an ecological community. Thus, pheromones generate structure in a food web and play critical roles in maintaining natural systems. Fear pheromones in humans. Evidence of chemosensory alarm signals in humans has emerged slowly, although alarm pheromones have not been physically isolated and their chemical structures have not been identified in humans so far. There is evidence for their presence. Androstenedione, for example, a steroidal, endogenous odorant, is a pheromone candidate found in human sweat, axillary hair and plasma. The closely related compound androstenone is involved in communicating dominance, aggression or competition. Sex hormone influences on androstenone perception in humans showed a high testosterone level related to heightened androstenone sensitivity in men, a high testosterone level related to unhappiness in response to androstenone in men, and a high estradiol level related to disliking of androstenone in women. A German study from 2006 showed when anxiety-induced versus exercise-induced human sweat from a dozen people was pooled and offered to 7 study participants, of 5 able to olfactorily distinguish exercise-induced sweat from room air, 3 could also distinguish exercise-induced sweat from anxiety-induced sweat. The acoustic startle reflex response to a sound when sensing anxiety sweat was larger than when sensing exercise-induced sweat, as measured by electromyograph analysis of the orbital muscle, which is responsible for the eye blink component. This showed for the first time that fear chemosignals can modulate the startle reflex in humans without emotional mediation. Fear chemosignals prime the recipient's defensive behavior prior to the subject's conscious attention on the acoustic startle reflex level. In analogy to the social buffering of rats and honeybees in response to chemosignals, induction of empathy by smelling anxiety of another person has been found in humans. A study from 2013 provided brain imaging evidence that human responses to fear chemosignals may be gender-specific. Researchers collected alarm-induced sweat and exercise-induced sweat from donors, extracted it, pooled it and presented it to 16 unrelated people undergoing functional brain MRI. While stress-induced sweat from males produced a comparably strong emotional response in both females and males, stress-induced sweat from females produced a markedly stronger arousal in women than in men. Statistical tests pinpointed this gender specificity to the right amygdala and strongest in the superficial nuclei. Since no significant differences were found in the olfactory bulb, the response to female fear-induced signals is likely based on processing the meaning, i.e. on the emotional level, rather than the strength of chemosensory cues from each gender, i.e. the perceptual level. An approach avoidance task was set up where volunteers seeing either an angry or a happy cartoon face on a computer screen pushed away or pulled toward them a joystick as fast as possible. Volunteers smelling a non-rostedinone, masked with clove oil scent responded faster, especially to angry faces, than those smelling clove oil only, which was interpreted as a non-rostedinone-related activation of the fear system. A potential mechanism of action is that androstenedione alters the emotional face processing. Androstenedione is known to influence activity of the fusiform gyrus, which is relevant for face recognition. Management. Pharmaceutical. A drug treatment for fear conditioning and phobias via the amygdala is the use of glucocorticoids. In one study, glucocorticoid receptors in the central nuclei of the amygdala were disrupted in order to better understand the mechanisms of fear and fear conditioning. The glucocorticoid receptors were inhibited using lentiviral vectors containing Cre recombinase injected into mice. Results showed that disruption of the glucocorticoid receptors prevented conditioned fear behavior. The mice were subjected to auditory cues which caused them to freeze normally. However, a reduction of freezing was observed in the mice that had inhibited glucocorticoid receptors. Psychology. Cognitive behavioral therapy has been successful in helping people overcome their fear. Because fear is more complex than just forgetting or deleting memories, an active and successful approach involves people repeatedly confronting their fears. By confronting their fears in a safe manner, a person can suppress the fear-triggering memories or stimuli. Exposure therapy has known to have helped up to 90% of people with specific phobias to significantly decrease their fear over time. Another psychological treatment is systematic desensitization, which is a type of behavior therapy used to completely remove the fear or produce a disgusted response to this fear and replace it. The replacement that occurs will be relaxation and will occur through conditioning. Through conditioning treatments, muscle tensioning will lessen and deep breathing techniques will aid in detensioning. Other treatments. There are other methods for treating or coping with one's fear, such as writing down rational thoughts regarding fears. Journal entries are a healthy method of expressing one's fears without compromising safety or causing uncertainty. Another suggestion is a fear ladder. To create a fear ladder, one must write down all of their fears and score them on a scale of 1 to 10. Next, the person addresses their phobia, starting with the lowest number. Finding solace in religion is another method to cope with one's fear. Having something to answer your questions regarding your fears, such as what happens after death or if there is an afterlife, can help mitigate one's fear of death because there is no room for uncertainty as their questions are answered. Religion offers a method of being able to understand and make sense of one's fears rather than ignore them. Society and culture. Death. The fear of the end of life and its existence is, in other words, the fear of death. The fear of death ritualized the lives of our ancestors. These rituals were designed to reduce that fear. They help collect the cultural ideas that we now have in the present. These rituals also help preserve the cultural ideas. The results and methods of human existence had been changing at the same time that social formation was changing. When people are faced with their own thoughts of death, they either accept that they are dying or will die because they have lived a full life, or they will experience fear. A theory was developed in response to this, which is called the terror management theory. The theory states that a person's cultural worldviews will mitigate the terror associated with the fear of death through avoidance. To help manage their terror, they find solace in their death-denying beliefs, such as their religion. Another way people cope with their death-related fears is pushing any thoughts of death into the future or by avoiding these thoughts altogether through distractions. Although there are methods for one coping with the terror associated with their fear of death, not everyone suffers from these same uncertainties. People who have lived a full life typically do not fear death because they believe that they have lived their life to the fullest. Religion. Religions are filled with different fears that humans have had throughout many centuries. The fears aren't just metaphysical but are also moral. Death is seen as a boundary to another world. That world would always be different depending on how each individual lived their lives. The origins of this intangible fear are not found in the present world. In a sense, we can assume that fear was a big influence on things such as morality. This assumption, however, flies in the face of concepts such as moral absolutism and moral universalism, which would hold that our morals are rooted in either the divine or natural laws of the universe, and would not be generated by any human feeling, thought, or emotion. From a theological perspective, the word fear encompasses more than simple fear. Robert B. Strimple says that fear includes the convergence of awe, reverence, adoration. Some translations of the Bible, such as the New International Version, sometimes replace the word fear with reverence. Fear in religion can be seen throughout the years. However, the most prominent example would be the Crusades. Pope Urban II allowed for Christian mercenary troops to be sent on a mission in order to recover the Holy Lands from the Muslims. However, the message was misinterpreted and as a result, innocent people were slaughtered. Although the Crusades were meant to stay between the Muslims and the Christians, the hate spread onto the Jewish culture. Jewish people who feared for their lives gave into the forced conversion of Christianity because they believed this would secure their safety. Other Jewish people feared betraying their God by conceding to a conversion and instead secured their own fate, which was death. It can also be argued that Christians feared their religion not being the primary religion, and this is why they committed mass murder. Manipulation. Fear may be politically and culturally manipulated to persuade citizenry of ideas which would otherwise be widely rejected or dissuade citizenry from ideas which would otherwise be widely supported. In contexts of disasters, nation-states manage the fear not only to provide their citizens with an explanation about the event or blaming some minorities, but also to adjust their previous beliefs. Fear can alter how a person thinks or reacts to situations because fear has the power to inhibit one's rational way of thinking. As a result, people who do not experience fear are able to use fear as a tool to manipulate others. People who are experiencing fear seek preservation through safety and can be manipulated by a person who is there to provide that safety that is being sought after. When we're afraid, a manipulator can talk us out of the truth we see right in front of us. Words become more real than reality. By this, a manipulator is able to use our fear to manipulate us out the truth and instead make us believe and trust in their truth. Politicians are notorious for using fear to manipulate the people into supporting their will. Through keywords and key phrases such as, "it is for your safety," or "it is for the safety of this country," politicians invoke fear within people that their safety is at stake, and people will ultimately follow in line in order for their safety to be restored. Fiction and mythology. Fear is found and reflected in mythology and folklore as well as in works of fiction such as novels and films. Works of dystopian and apocalyptic fiction convey the fears and anxieties of societies. The fear of the world's end is about as old as civilization itself. In a 1967 study, Frank Kermode suggests that the failure of religious prophecies led to a shift in how society apprehends this ancient mode. Scientific and critical thought supplanting religious and mythical thought as well as a public emancipation may be the cause of eschatology becoming replaced by more realistic scenarios. Such might constructively provoke discussion and steps to be taken to prevent depicted catastrophes. The story of the youth who went forth to learn what fear was is a German fairy tale dealing with the topic of not knowing fear. Many stories also include characters who fear the antagonist of the plot. One important characteristic of historical and mythical heroes across cultures is to be fearless in the face of big and often lethal enemies. Athletics. In the world of athletics, fear is often used as a means of motivation to not fail. This situation involves using fear in a way that increases the chances of a positive outcome. In this case, the fear that is being created is initially a cognitive state to the receiver. This initial state is what generates the first response of the athlete. This response generates a possibility of fight or flight reaction by the athlete, which in turn will increase or decrease the possibility of success or failure in the certain situation for the athlete. The amount of time that the athlete has to determine this decision is small, but it is still enough time for the receiver to make a determination through cognition. Even though the decision is made quickly, the decision is determined through past events that have been experienced by the athlete. The results of these past events will determine how the athlete will make his cognitive decision in the split second that he or she has. Fear of failure, as described above, has been studied frequently in the field of sports psychology. Many scholars have tried to determine how often fear of failure is triggered within athletes, as well as what personalities of athletes most often choose to use this type of motivation. Studies have also been conducted to determine the success rate of this method of motivation. Murray's exploration in Personal was one of the first studies that actually identified fear of failure as an actual motive to avoid failure or to achieve success. His studies suggested that in avoidance, the need to avoid failure was found in many college-aged men during the time of his research in 1938. This was a monumental finding in the field of psychology because it allowed other researchers to better clarify how fear of failure can actually be a determinant of creating achievement goals as well as how it could be used in the actual act of achievement. In the context of sport, a model was created by R.S. Lazarus in 1991 that uses the cognitive motivational relational theory of emotion. It holds that fear of failure results when beliefs or cognitive schemas about aversive consequences of failing are activated by situations in which failure is possible. These belief systems predispose the individual to make appraisals of threat and experience the state anxiety that is associated with fear of failure in evaluative situations. Another study was done in 2001 by Conroy, Poshwardowski, and Henshin that created 5 aversive consequences of failing that have been repeated over time. The 5 categories include experiencing shame and embarrassment, devaluing one's self-estimate, having an uncertain future, important others losing interest, upsetting important others. These 5 categories can help one infer the possibility of an individual to associate failure with one of these threat categories, which will lead them to experiencing fear of failure. In summary, the two studies that were done above created a more precise definition of fear of failure, which is a dispositional tendency to experience apprehension and anxiety in evaluative situations because individuals have learned that failure is associated with aversive consequences. Inability to experience fear. People who have damage to their amygdalae, which can be caused by a rare genetic disease known as Urbach-Wiethe disease, are unable to experience fear. The disease destroys both amygdalae in late childhood. Since the discovery of the disease, there have only been 400 recorded cases. This is not debilitating, however, a lack of fear can allow someone to get into a dangerous situation they otherwise would have avoided. For example, people who possess a lack of fear would approach a known venomous snake while those who possess fear would typically try to avoid it. Fear is an important aspect of a human being's development. Another possibility of a lack of fear could be antisocial personality disorder, or more commonly known as sociopathy, which is a mental health condition where people often manipulate others. People who suffer from this disorder lack a physiological response and are capable of lying or not experiencing fear. When a person is approached by something they fear or has a phobia, the body will react in a way that conveys this fear. For example, a person who possesses arachnophobia, or a fear of spiders, would experience a physical response such as nausea, dizziness, or even shaking when approached by a spider. However, a person with antisocial personality disorder lacks this capability and would not activate their fight or flight response to such fear. They also show a disregard for the safety of others, and may intentionally harm them or allow harm to come to them.